A first look at cells. Most plant and animal tissue is made up of different units called cells. These were first seen by Robert Hooke when he looked at a piece of cork. Within each plant or animal, there are many different types of cells, each doing a different job. They are all important to the process of keeping the plant or animal alive. Preparing cells. Normally, when scientists want to look at cells from a plant or animal, they cut off a very thin slice called a section. See pages 24 to 25. However, you can look at plant cells from an onion and animal cells from your cheek without making a section. On pages 30 to 31, you can find out how to stain samples to see them better, but you can start off by looking at them as they are. You should still see the main features. Remove the brown skin from the outside of an onion and cut out a piece from a layer of the inside flesh as shown in the diagram. On the inside, you will find a very thin, almost transparent membrane or skin. Peel this away using a pair of tweezers and make a temporary mount by following the instructions in the panel below. Look at the slide using bottom lighting. The cells will be almost transparent. Layer of onion flesh. Peel membrane off. Membrane under low power. Individual cells. You may be able to see a roundish patch in each cell. This is the nucleus, the cell's control center. Lightly scrape the handle of a clean, disinfected spoon around the inside of your cheek. Carefully wipe the scrapings onto the center of a slide and spread them out gently with the edge of another slide. Leave the cells to dry, or dry them by waving the slide in the air, and then make a temporary mount. Examine the slide using bottom lighting. Never take cells from anyone's cheek but your own. Spread the cells with the edge of a slide. This is called a smear. Cheek cells viewed under low power. Animal cells are less regular in shape than plant cells. You may be able to see the nuclei again. After use, always disinfect slides, seal them in a plastic bag and throw them away. Making a temporary mount. Most biological specimens have to be kept wet, otherwise they will dry up and decay. If the specimen is only going to be kept for a short time, a temporary or wet mount is used. You can find out how to make a semi-permanent mount on page 36. Place a drop of water on a clean slide using a pipette or glass rod. Carefully transfer the specimen into the drop of water. Pick up a cover slip by its edges and place one edge on the slide. Lower the other edge of the slip. The water will spread out under it. Soak up any excess with blotting paper. You may trap some air bubbles. They look like dark rings under the microscope. A few do not matter, but try again if there are lots. Special cells. You have seen that animal and plant cells are different by looking at onion and cheek cells. There are also many different types of plant and animal cell. These vary widely in size, shape, and appearance. This is because each type of cell is specialized to do a certain job within the plant or animal. There are two examples below. Specialized cells. Animal nerve cells carry electrical signals to and from the brain. The fiber of a nerve cell can be several meters long in large animals like whales. Cytoplasm. Nucleus. Pairs of guard cells on the underside of a leaf change shape to open or close holes through which the leaf breathes. See also page 27. Cytoplasm. Nucleus. Cell structure. In the experiments on the left, you could see the outline of the cell and perhaps the nucleus. With higher power, you would be able to see more features inside cells. The pictures below show the main differences between plant and animal cells. The nucleus is found in nearly all cells. It controls everything which happens in a cell. Animal cell. The contents of the cell, except for the nucleus, are called cytoplasm. This can contain a number of special things. Animal cells have granules of glycogen, which act as a food store. The cell membrane is a thin layer which holds in the contents but lets liquids and gases through. Plant cell. A cell wall is only found in plant cells. It is much thicker than the cell membrane and keeps the cell's shape. Chloroplasts are found in plant cells. They make food from sunlight and make leaves green. Plant cells also contain starch granules, which are a store of food. Many cells have fluid-filled spaces called vacuoles. In plant cells, they are large and help to keep the cell's shape. Cork cells. Robert Hooke was a 17th century microscopist. He examined cork and saw that it had a regular structure of units, which he called cells. See also pictures, page 4. You can see these cells if you look at a slice of a cork. Either just put a largest piece on the stage or make a temporary mount of a very small piece. Use top lighting. Cork cells. The cells are dead wood cells. Each has a cavity inside. See page 27 for pencil wood cells.